And then quite often we scale the data. We do a Z-score transformation before doing PCA or any other type of dimensionality reduction, where it's basically you do a linear transformation for each gene, where we set the mean to 0 and standard deviation to 1. So basically that means that each gene is equally important when we do a PCA. So once we've normalized our data, we selected our variable genes, then we might have other confounding factors in our data. Hopefully we now controlled for sequencing depth and dropout rates per cell. But we still might have cell cycle, cell size, and so on, different batches, different quality metrics that we need to remove. And a common way to do this is simple linear regression. So the idea is that you take your data and your variable of interest, you fit a line to this data and you calculate the residuals. And instead of taking the expression value, you take the residuals, and by that you should remove all the variants that are explained by this variable of interest. So this might be percent mitochondria that we know correlates strongly to the quality of the cells, or it might be the number of detected genes. And you can add multiple different factors into this regression model. There's a bunch of different other tools that are just common in different statistical packages and so on to remove batch effects. Like RNA-seq, COMBAT seems to work quite well, also for single cell data. But pretty much all of them, you need to think about that whatever you're doing and tweaking your data, it's not always the case that you will maintain all of your biological information when you start regressing out too many different factors. So I think it's always a good idea to do first an analysis with the data as it is, to look at what you get. I see a strong effect of this or that, and then you can start playing around with adding different factors that you regress out. And as I said, I would quite often start with like percent mitochondrial read and gene detection rate. If I know that I have a strong cell cycle dependence, I would remove that as well, if that's not what I'm looking for in my data. Batch effects you can remove with this linear regression, but there are a lot of issues if you don't have like perfectly matched populations between. But I think Paulo will talk more about that in his lecture. So there are better ways to remove the batch effects than doing regression. And then I think it's always a good idea to check these QC parameters like mitochondrial reads and so on before and after you do these regressions. And also think about, I mean, if you remove cell cycle, for instance, and the whole study is about some differentiation process where cells goes from cycling to non-cycling cells, you might actually remove the whole differentiation path by removing cell cycle. So it's not easy to really know what you should remove and not. So in practice, it's very easy to run this. In Seurat, scale data runs both the Z-score transformation and you add in these variables vars to regress with whatever you want to regress out, and you can select linear or Poisson or negative binomial regression. In SCRAN, there's no automatic centering and scaling in the PCA step. Instead, there are slightly different ways of doing this, because they do this decomposition into technical and biological variants, where you basically define your technical variance in these block factors. And then you denoise your PCA to remove these block factors. And in ScanPy, it's the regress out and scale functions.
Which one to use? Like in the Seurat package, there's so many different. What's the, I mean, often it gives similar information, but it's difficult to choose which.
I haven't really done a thorough evaluation of them. My feeling is, like you say, I get very similar results. It's also quite often the case when you do this kind of analysis that you don't really know what the ground truth is.
But one comment that I can give is that the linear one is much faster than the other two, because it uses the already transformed data, while Poisson negative binomial go back to the counts and create a whole model from the beginning. So that's why actually the linear is the default.
Great.
And also what it actually does is, if you do variable gene selection first, and then you only run scale data on the variable genes, which also speeds up quite a lot, because then maybe you have 3,000 genes instead of 30,000.